DNA microarrays are molecular biology tools used to measure the expression of thousands of genes simultaneously. They consist of a glass slide or silicon chip containing thousands of DNA probes arranged in a grid. These probes hybridize to complementary nucleic acid sequences in a sample, allowing researchers to determine which genes are expressed and at what relative levels. Microarrays can be classified into two main types: cDNA arrays or gene arrays and oligonucleotide arrays. cDNA arrays contain relatively long DNA fragments derived from cloned genes, producing strong and sensitive hybridization signals. For reliability, the same cDNA probe is often replicated at multiple spots on the array. In contrast, oligonucleotide arrays contain short synthetic DNA probes, typically 20 to 70 nucleotides long. A single gene is often represented by multiple probes targeting different regions of the gene, improving the accuracy and specificity of expression measurements. Oligonucleotide arrays can also distinguish between highly similar gene sequences, transcript variants, and mutations. The microarray procedure begins with the extraction of total RNA from cells or tissues. Because the abundance of messenger RNA reflects the expression levels of protein-coding genes, mRNA molecules are selectively reverse transcribed into complementary DNA using oligo-DT primers and reverse transcriptase. In some protocols, mRNA is first purified from the total RNA before cDNA synthesis. Fluorescent labels are incorporated into the cDNA during synthesis or attached afterwards depending on the labeling method used. The labeled cDNA is then denatured to produce single-stranded molecules. Depending on the microarray platform, the labeled nucleic acids may also be fragmented to improve hybridization efficiency. In some microarray platforms, the array may undergo pre-hybridization treatment to reduce non-specific binding and background fluorescence. The labeled sample is then mixed with hybridization buffer and applied to the array, where target nucleic acids hybridize to complementary probes. Hybridization is typically performed in a specialized chamber at 45 to 65 degrees Celsius for 16 to 24 hours to allow specific probe target binding. Following hybridization, the array is washed to remove unbound and weakly bound molecules. Initial washes remove excess sample, while more stringent washes performed at controlled temperatures eliminate mismatched hybrids, ensuring that only highly specific probe-target interactions remain. The array is then dried and scanned using a laser fluorescent scanner. The laser excites the fluorescent labels and detectors measure the emitted light from each spot. Specialized software converts the fluorescence image into quantitative data by measuring signal intensity, subtracting background noise, and normalizing the results. The fluorescence intensity of each spot reflects the expression level of the corresponding gene. Microarray procedures may be performed using either a one-color or a two-color approach. In a one-color microarray, RNA from a single sample is converted into fluorescently labeled cDNA and hybridized to an array. The fluorescence intensity at each spot indicates the expression level of the corresponding gene. To compare two samples, such as healthy and diseased tissue, separate arrays are hybridized with labeled nucleic acid from each sample, and the resulting expression profiles are computationally normalized and compared. In a two-color microarray, RNA from two samples are converted into cDNA and labeled with different fluorescent dyes, commonly represented as red and green. The labeled samples are mixed and hybridized to the same array. After scanning, the color of each spot reflects the relative expression levels in the two samples. Spots that appear predominantly red indicate higher expression in one sample, whereas predominantly green spots indicate higher expression in the other sample. Yellow spots indicate similar expression levels, while intermediate shades such as orange or yellow-green represent varying degrees of differential expression. Dark spots indicate little or no expression in either sample. Pause the video here to attempt the quiz question. In conclusion, microarrays are powerful tools for large-scale gene expression analysis. Compared to northern blotting and RT-PCR, they can analyze thousands of genes simultaneously. Although RNA sequencing provides a more comprehensive and sensitive analysis of the transcriptome, microarrays remain cost-effective tools for comparing healthy and diseased tissues, identifying biomarkers, investigating drug responses, and studying gene regulation. As answering questions help reinforce learning and improve retention, do attempt the following questions. If you found this video useful, please press the like button and subscribe to receive notifications of upcoming videos. Thank you for watching.